This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 613, recorded on May 13th, 2020. I'm Vincent Yellow, and you're listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody. It was 32 degrees when I woke up this morning, so that's 0 Celsius. And right now it's 49, which is 9 Celsius, and it's going to get up to 61, which is 16.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We got 70 degrees and cloudy, going up to a high of 85. Yesterday, it rained like crazy. We got over 2.5 inches of rain, which is just awesome. I love it.
It's 13 Celsius here. It's sunny. Well, it is chilly at night. You're right. Absolutely right. If you like what we do here on TWiV, and I know many of you do, many of you are new to TWiV, consider supporting us. Microbe.tv/contribute. So we will make some inroads on email today, but first we have a few stories we'd like to chat about with you or to you, because you can't chat with us, although we can chat amongst each other. Kathy, tell us about this Mother Jones article.
Oh, I didn't know we were going to talk about that, but it's the title is "Science Has an Ugly, Complicated Dark Side, and the Coronavirus is Bringing It Out." And the gist of it is that there's a lot of pseudo experts that are weighing in. And even scientists whose field is not necessarily coincident with what they're opining about in their writings. There's a related story in Wired. So it's the title of that is "Don't Be Cooled by COVID-19 Carpetbaggers." And I think the Wired story had 3 criteria for how you should evaluate a news thing, and it's loading slowly, so maybe just check it out.
I think I got it here. Transparency and contributions. So you got to be able to track the references and et cetera. Participation in the scientific ecosystem. That's a little vague.
Yeah,
COVID-19 experts should make an attempt to participate in the existing ecosystem of science, where findings are produced and shared in a forum of scientific preprint or manuscript. In other words, not just your thing stuck out there on some server somewhere. And the other is transparency about motivations and methods. At any rate, there is, real science takes time. And as we have pointed out many times before, is self-correcting. There's always bad science going on, but it winds up falling by the wayside because people are trying to confirm things all the time, and it just takes time. Everything now in the COVID area is compressed and amplified in the media, and so the bad science winds up getting more notice than ordinary.
Mm-hmm. Yeah,
got to be careful.
Yep.
Got to be careful.
This always happens when there's a outbreak of some kind. All the bad stuff gets amplified, and Zika and Ebola, flu pandemics. The same thing happens. You have lots of preprints. You have lots of new people coming into the field. You have lots of people commenting on it, and this is what's happening here. Now, the preprints are a big issue because they're not reviewed, and a news source picks it up and they don't know what to make of it. They get a few comments from people who may not know about it, and it goes crazy. And many of those don't see the light of day. Unfortunately, in this situation, Nature, Cell, and Science and many other journals want to publish about SARS-CoV-2. So in my opinion, they are less stringent about their requirements, and things get published that should not be in those supposed luxury journals where they claim, they have the cream of the science, which we've argued in the past is not always true. But they publish stuff that just is meant to get attention, and I just object. But that's the ecosystem we're in, and my little voice is not going to do anything about it.
Vincent, you'll appreciate this. I was talking to one of my students who I was interviewing, so to speak, for his med school letter of reference. And I told him how I had done a screenshot for next year's slides to put on the evolution lecture slide about the virus is mutating. And he laughed. He got it immediately. So I evidently made a good enough point of it during class.
Good.
The preprint servers are kind of a special issue because, they are not peer reviewed, but I personally am glad they're there so that there's at least something that, this some of it, maybe much of it, is quite good. And it happens really quickly. The problem is you have to be able to discriminate. That's part of the reason we're here.
Yeah, I think you ought to listen to TWiV when you want to know about viruses, okay? Not enough of you are listening. Christian Drosten gets over 1.5 million downloads per episode. We're not even close. I was just talking to a reporter in Germany about that. And, don't get your science from the news. They don't know what they're talking about. I mean, they call us to get things explained. And so here last week, this spike mutation, spike amino acid change, was making the rounds. And a reporter called. She seemed to get what I was explaining, and she wrote her article, and it looked pretty good. Then her last quote, the kicker, as they say, they want to end up with something profound, is a doctor somewhere, who says, "We don't know what this means at this time." And I wrote to her, "Why did you put that at the end? You negate everything that we said about mutations not being meaningful until proven so. Why do you put this guy's kicker at the end?" She said, "Oh, I'll get back to you on that." And I never heard from her.
Yeah, right. She wants to leave the door open, right? It's like a headline with a question mark, a headline with a false statement. Could this be happening?
Yes. So that's where we are. What's tomorrow? Sunday, May 17th.
Tomorrow is Thursday, May 14th.
Oh, sorry.
What day is it, Vincent?
What is it? Smallpox vaccination day?
Smallpox vaccination day. That's the day that Edward Jenner took lymph from a lesion on the hand of the milkmaid Sarah Nelms and used it to inoculate or vaccinate James Phipps, 12-year-old boy.
What's lymph?
Pus, dude.
Okay. Okay. Just, some people may not know what it is.
That's right. She had a blistering lesion, which was cowpox. She had cowpox, this skin disease acquired from cattle that Jenner's investigations had indicated to him conferred immunity to smallpox. And so he did the experiment. He took material from the lesion on this milkmaid's hand and put it into the arm of a 12-year-old boy using established techniques that were used using actual smallpox variolation, which, was effective but had like a 1% mortality rate. That's better than 30%, but it's not great. At any rate, and then he did the what seems in this day outrageous thing of like 12 days, I think, later or something like that.
Mm-hmm.
He followed up with the procedure I just described, variolation. That is, he challenged the kid with smallpox, and the kid James got no reaction. So that was anecdotal, but indicating that his procedure had conferred immunity to smallpox. And that was the beginning of immunology, the beginning of vaccinology, the beginning, even though we didn't know it, of virology. All very cool.
And we will not, in my opinion, do challenge experiments with SARS-CoV-2 vaccines because they're too dangerous. And we have tons of people writing in, "Don't you want to save thousands of lives?" No. So you want to sacrifice someone else's life for that? I don't think that's appropriate. Now, if the powers that be decide, fine. But I don't think we ought to do it. And this is my podcast, our podcast, and we can say what we want. Not always right. I don't think we should challenge people where they could die of the challenge. That's crazy.
Right. We're going to come to a paper later that does address the challenge trials with another reference that I didn't have time to look up yet.
Kathy, they really have this cow's hide somewhere?
Yeah. I found it. I sent the information to Rich, and he said, I don't know if that's really true. So then I Googled it and found it and sent him the picture and said, now we don't have to go.
I'm still skeptical. I've been dealing with this stuff for over 40 years, and I've never heard about Blossom the cow's hide. To me, it's kind of like the Shroud of Turin or something like that. This is some sort of religious artifact. I'm not sure this is the real thing.
It says it's in the St. George's Archives and Special Collection at the University of London.
I probably ought to write to Jeff Smith about this and say, okay, what's this up? What's up with Blossom?
Yeah, that's good. It's good. He would explain it. That would be fun.
Yeah.
And it's the Jenner's birthday is coming up too, right?
That's what's on Sunday, May 17th. Yes.
Yeah. Wow. All right. Speaking of vaccines, I wanted to chat a bit about this. Maybe, in each episode we'll cover a particular vaccine. We talked a bit about the mRNA vaccine that is being tested via Moderna. And so today, the Oxford Vaccine Institute, is that the name of it? University of Oxford.
Jenner Institute.
Look at that. We talk about Jenner and we flow right into Jenner Institute. And Oxford, they have a vectored vaccine that is doing interesting things, and we'll talk about that. So let's do that. So I have an article that I don't know where I got it, but it's in Biospace. It's called Jenner Institute's COVID-19 vaccine expected to be tested in 6,000 people starting in May. And of course, this is already out of date because the testing has already started, but it gives an interesting background on the vaccine, how it got started, how they decided, they were working on a bunch of different vaccines at the Institute. And they decided when SARS-CoV-2 started circulating to focus everything on this using a platform they'd established, which we'll talk about. But I just want to point out here, and this will come back later, they have not yet reached a deal with a North American manufacturer. And I don't know if this is still true, partly because major pharma companies generally require exclusive worldwide rights before investing in a possible therapy. And Adrian Hill, the director of the Institute, said, I personally don't believe that in a time of pandemic, there should be exclusive licenses. Nobody's going to make a lot of money off this. And I think that's great. So just get with it, North American manufacturers.
Yep.
Save some lives.
Yep. This is a time for everybody to be all in.
So let's, Kathy, tell us, this is an interesting vectored vaccine.
Right. So
tell us a little bit about it.
Yeah, we've talked in the past just a little bit about adenovirus vectored vaccine. So adenovirus is what I've worked on, and I have always said it's one of the common cold viruses. It's not one of the major ones that you hear about so much, but it's a DNA virus that you can use to insert genes of interest. In this case, it would be the coronavirus spike protein. Adenoviruses are quite immunogenic, and the way that they have made this vector takes advantage of the fact that you can use a interesting selection mechanism to rapidly produce your vectors. So adenovirus mutants or vectors have been made sort of a long-standing traditional way, but then around 2001, a technique called recombinering was pioneered, and I can't remember the exact year when it was used for first for adenoviruses, but recombinering is a term that I'm trying to find my notes here. Stands for, or is a portmanteau, I think, is the way that word form is called. No, portmanteau is a word inside a word. Anyway, recombination mediated genetic engineering. So recombinering. And so the vector is one where you put the entire adenovirus genome into a bacterial plasmid, and then you can propagate it in bacteria. And the reason this is important is that it's really hard to work with large vectors, and this is going to be on the order of a 30 to 40 kb adenovirus genome and a 4 to 5 minimum size plasmid. So you're talking something that's pretty big for the bacteria to handle. So this method was pioneered by Don Court at the NCI. And the idea is that you can use a positive and negative selection to generate your vector. So you use GalK, which is a bacterial gene, and you engineer your vector and put in the GalK, and you can select for the vectors, the plasmids and the bacteria that grow because they have a plasmid with the GalK. So you do that selection on medium that only has galactose, and you can only get plasmids growing if they got that gene. So you can select for the galactose. So in this case, they're going to delete the E3 region of the adenovirus. And I forgot to mention early on that this is the chimpanzee adenovirus. So they're going to delete E3, and so they put the GalK into the E3 region and obliterate the whole E3 region. And they do this for early region 4 as well. And in the case of early region 4, then they use the original recombinering method, which is that you can also then select against the GalK insertion with a particular drug that makes a toxic product if the GalK is there. So things don't survive if they have that. And so you use that counter selection or selection against the GalK to put in your gene of interest, which might just be a modified early region 4 gene or some other gene. So they start out with a vector that they do by engineering. In this case, they delete early region 1, and that makes it replication incompetent. And then they do this E3 deletion by doing the GalK insertion. But in fact, they put restriction sites on that. And so to get rid of the E3, they just do a restriction digest. But then they also do the E4 recombinering with the GalK plus and minus selection. And the reasons to, as I said, early region 1, you delete so that it's recombination defective. Replication defective. Early region 3 gives you space to clone in your gene of interest, about 8 kb in this case. And the early region 4 part is interesting in this case because they know from past experience that if you include early region 4, having early region 4 there is important for the growth of the virus. And they're doing a chimpanzee adenovirus, but they're going to be growing it in human 293 cells, which express human adenovirus E1b proteins and E1a. And to get the best advantage of having the E4 proteins, they need to have the homologous human adenovirus E4, and they put in E4 ORF6 through this recombinering mechanism to increase the yield of their vector. And then later, they find that they get even better results if they also include ORF6, ORF7, and ORF67. These are open reading frames. This is how the genes are called. Sorry. That's just the way it is. And also the chimpanzee virus, E4, ORFs 1, 2, and 3. So all of this is to make a really optimized vector that they can propagate in these human 293 cells. They also make a point that they use a single cell infectivity assay so that they can control for the different replication among the vectors that they want to compare. And this is an infectivity assay, but it's not a plaque assay. Instead, you plate out the cells at a relatively low density and infect them. And then if your vector has a fluorescent gene in it, like green fluorescent protein, you can screen for the cells that get the GFP. But ultimately, you don't want to have such a gene in your vaccine vector. So they use antibody-based assay for identifying infected cells that's recognizing the adenovirus structural protein hexon. And they also do additional controls with their chimpanzee vector to determine whether the transgene affects this infectivity assay, whether the E4 modifications that they put in affect immunogenicity, and whether there's a human seroprevalence against this particular chimpanzee vector. And so all of those controls are in place. And I kind of went through that without stopping or almost taking a breath.
That's cool.
You have a question?
Yeah, I have a number of questions. So I just kind of want to summarize and make sure I got this straight. So I assume that we're using a chimpanzee adenovirus because humans are unlikely, or probably don't, have any pre-existing immunity to the chimp virus, which might otherwise, if you used a human adenovirus, compromise the effectiveness of the vaccine, correct?
Correct. We have antibody to the primarily studied adenovirus 5 and 2, which are like kissing cousins. And so, although lots of vector work had been done in them early on, people started to realize that this was not going to work because of people's pre-existing immunity.
So later on, I want to come back to the pre-existing immunity thing, but I want to go through the rest of it. So the end result of all of this recombinant DNA technology is I have a chimp adenovirus that's missing the E1 gene, which is essential for replication. And has inserted in it a human E4 gene because, as I understand it, the complementing cell line, the thing on which you can grow this, provides human E1, and it needs a human E4 subunit to work best. Right?
Right. And that E4, if I understand it correctly, is in the position of the E4 of the chimpanzee. It's not going into E1. It's in the E4 region.
Right. And you say that E3 was deleted to make room for the transgene. Does the transgene that can be interpreted in a couple of ways? Either that the transgene goes into the E3 region, or it simply shortens the genome so that I've got more space for because there's going to be a size limit on how much I can put in this, I presume, based on the packaging capacity. So I could put the transgene in. Where does it go? Does it go in E1 or E3? Do you know?
That I don't recall whether it goes into E1 or E3.
All
but you're absolutely right about the packaging limit side and needing to have space for it.
All right. So I've got basically a crippled adenovirus with a transgene in it, and what's controlling transcription of the transgene, expression of the transgene? Some promoter from space?
From what I could tell, the details for this particular chimpanzee vector with either the MERS spike or the SARS-CoV-2 spike that we're thinking about, I can't quite tell from the literature, but the previous one uses human cytomegalovirus immediate early promoter, which is
that's basically a constitutive that is always on high expressing promoter from another virus, cytomegalovirus.
Right.
So I've got this crippled virus with a transgene in it, and that's not going to grow without E1, but I can grow it because I got cells that express E1, and because I got cells that express E1. Okay, cool.
Now, when you get this virus, this chimp ad with a transgene, in this case, the spike glycoprotein gene from SARS-CoV-2, and now when this infects human cells, what happens?
So the virus goes in and starts to replicate and express.
The DNA replicates?
Sorry, I misspoke. The virus goes in and there will be gene expression. And so you will get gene expression of the transgene and the E4 genes that are there, some of them chimpanzees, some of them human adenovirus genes. Right.
Okay. And so the protein will be made, and then you will not get new adenovirus particles made, right?
Right. You should not.
Probably, you don't even get adenovirus DNA. You shouldn't even get adenovirus DNA replication, correct? Because you need E1 is essentially a transcriptional activator that turns on all of the genes required for DNA replication. And so if it's not there, that's not going to happen. Is that correct?
Correct.
Okay. So you're going to get minimal transcription, a couple of adenovirus genes and the transgene.
Right.
And there should be no viral DNA replication, no viral capsids protein synthesis, which I think is important for these vector immunity considerations. It should be just really primarily the transgene and a couple of adenovirus genes that are not going to do much.
Yeah. I wanted to make that clear for listeners who want to understand when humans are vaccinated, the viruses will go into cells and make proteins, but that you're not going to make new adenoviruses. So whatever dose you get is going to give you whatever immunity happens. You're not going to amplify it.
By the way, just a little aside here, this whole viral vectored vaccine technology is relatively novel. Okay. Vincent, you pointed out there are 2 licensed viral vectored vaccines, right?
Correct. Does anyone know what they are? I'll give our listeners a few seconds to write them down.
I got it wrong. At any rate, and there are a bunch of these that are in use in veterinary practice. In particular, I'm familiar with poxvirus vectored vaccines, in particular canary pox, a fowl pox, a bird pox virus that expresses the rabies glycoprotein is used to vaccinate animals against rabies. And I think there are several others as well, but I can't detail them.
But humans there too, right?
Yeah, in humans there are just 2.
Yeah, the Ebola vaccine, Ervebo, is a Ebola glycoprotein in a vesicular stomatitis virus background. And then Dengvaxia, which is the dengue virus structural proteins in a yellow fever vaccine backbone. Those have been licensed for use in humans. But there, as you said, there are animal vectored vaccines already used in animals.
And if you look at the document that we have cited previously from the World Health Organization that keeps track of vaccines that are under development, there are many of these viral vectored vaccines being considered, more than one in clinical trials. Actually, I ought to get out the latest and I can come back to it. So this is I would call it an emerging technology that we're going to learn a lot about in the course of this pandemic.
Yeah. So this is this platform, this chimp adenovirus that Kathy described, has been used by the Jenner Institute in collaboration with others to make a vaccine candidate against MERS coronavirus. And they have, so I have 2 references to put in the show notes. One is a bioarchive preprint, a single dose of, so these are called ChAdOx, chimpanzee adeno Oxford ChAdOx, just rolls off the tongue. It's pretty good. ChAdOx MERS. So this ChAdOx1 MERS provides broad protective immunity against a variety of MERS-CoV strains. So these are using non-human primates, rhesus macaques, who are immunized a single intramuscular vaccination with the ChAdOx1 MERS, and then they're challenged and they're protected against respiratory injury and pneumonia, reduction in virus load. Okay, so that's a human vaccine candidate tested in non-human primates. And that of course, the challenge is done at Rocky Mountain Labs over in Hamilton, Montana, where they have an NIH lab that can do these sorts of things. So that's the one thing. And then this same vaccine was used in a phase 1 trial in humans. This was just published last month. And safety and immunogenicity of a candidate MERS coronavirus viral vectored vaccine, dose escalation, open label, non-randomized, uncontrolled phase 1 trial.
They really put it all out there.
It's all there. And so they get several different doses. Humans, 3 different doses, and they give them these, and they follow side effects, and they seem to be well tolerated. And then they find immunity. They look at the antibodies that are made as well. And so this is a satisfactory outcome. And that's why this one, I think, SARS-CoV-2 version is going into people so quickly, because we have this experience with a ChAdOx MERS, right?
Right. And they followed the people in this trial for up to 12 months. So this really started a while back.
Now, this means it's good in a non-human primate model, the MERS vaccine. It's safe and tolerated well and seems to generate antibodies in people. For MERS, now they've made, of course, a SARS-CoV-2 version. And that is actually has been, so now this is the subject of a couple of letters that we have, which are quite informative. I
think there were 1 or 2 more things that we may have wanted to say. Oh, because Rich had a question about, he did ask how the transgene transcription is controlled. Mm-hmm. He asked how much gene expression or replication is there in the human host. We've talked about the fact that there's gene expression, but shouldn't be viral replication. And then, is there an immune response to the adenovirus vector? And does this affect boosting or repeated use of the platform? And yes, there probably is an immune response to the adenovirus vector. It's being put in at pretty high doses of virus particles. And so those antigenic proteins on the vector's surface from adenovirus are going to raise immunity. And so, yes, in this Lancet paper, they say, as Vincent mentioned, that they did get immune response. They measured neutralizing antibodies to the MERS coronavirus and cell-mediated immunity, and they saw both of those. Now, importantly, this trial was a single-dose immunization, and that gets at the question about whether repeated use is going to be a problem. And most likely it will be, because adenovirus vectors have the problem that there's rapid development of adenovirus vector immunity. And that poses a significant challenge to using these as vectors if you want to boost with them. So a single dose immunization being successful, being efficacious is important, because you're not going to be able to go back in.
And this would apply either to a boost or to, let's say, I mean, if these viral vectored vaccines become acquire more common usage, then if you want to use the same platform to immunize against another disease, there may be a problem if you've already had a vaccine from the same platform to some other disease.
Correct.
Now, I'm sure that this is a big issue in all of the developers. So there may be something about this chimp platform that gets around this. Maybe somebody who knows the details can write into us and comment on this issue, because I think it's important.
Yeah. As I said, in one of these studies, they did look at controlling for whether there was what the seroprevalence was. And yeah, it was in the July 2012 PLOS One paper that includes these people from Oxford. And results are shown, and they're basically, if I read it quickly here, they're not seeing cross-reactivity or high seroprevalence in humans against this chimpanzee adenovirus. So
you wouldn't think so unless there was a lot of cross-reactivity or something.
Yeah. Okay. Yeah. They used 2 previously uncharacterized panels of human sera. The values are 0% in UK adults and 9% in Gambian adults. Okay. So that.
Wow. Yeah. That's interesting.
Yeah.
Yeah.
Wow. That's very interesting.
Guess you have some contact with.
Yeah. Holy cow. That's really interesting.
Yeah.
So it's worth pointing out that with human adenovirus platforms, of which there are several around, this can be a problem. Ad5 was used as a vector for the HIV glycoprotein in an AIDS trial. We've discussed this before, and that actually resulted in an enhancement of disease because the pre-immunity to the Ad5 vector, when you got the vaccination, stimulated the production of T cells to the ad vector because of the pre-existing immunity, and those are exactly the cells that the HIV wants to infect. At least that was the explanation that I recall for really an enhancing effect of the vaccine. They stopped that trial. By the way, I just looked back and I count on the tally of vaccination trials ongoing about 19 different viral vector vaccines, 2 of which are actually in clinical trials. Both of those are adenovirus. One is an Ad5 platform, and the other is this chimp adenovirus platform. The others can be divided into replicating and non-replicating viral vectors. The non-replicating viral vectors, the vast majority of them are either adenovirus or MVA, which is a crippled pox virus. Actually, it's a crippled pox vaccinia virus, and then a couple of replicating viral vectors, including measles and vesicular stomatitis virus. So we're going to learn a lot from this.
So I did refer just now to this PLOS One paper, which I'm not sure Vincent was planning to put into the show notes, but I think it's worth it for people who really want to get into the weeds on this. It's a novel chimpanzee adenovirus vector with low human seroprevalence, improved systems for vector derivation, and comparative immunogenicity.
Yeah, we'll put that in.
And that was all the way, is that the one from 2012?
Yes.
Okay, so the point being that this has been going on for a long time.
Yes.
So this discussion was prompted by an email from Brian and Jan, who it's mother and son, I presume.
Yeah, I think so.
She says, my mother and I have become big fans of TWiV, and they want to know about this Oxford vaccine, and they thought it was more promising than we had said. You're focusing a lot on Remdesivir, but why don't you talk about this vaccine? So that's why we did talk about it, and that the next one is quite interesting. Maybe Kathy, you can read Brian's.
Sure. Brian writes, "Hi, Vincent et al. It has been a sunny weekend in the UK with highs of 24°C over the past 2 days, though set to change with a drop to 14°C on Sunday. Heard you talking about combined phase 2-3 trials. Just wanted to correct and say that the UK ChAdOx and COVID-19 is actually a phase 1-2 combined trial. So phase 1 is safety and phase 2 is a small trial for efficacy, right? That's how you would define those trials. Okay.
Yep.
All right. And so he puts in links about those. And here's the interesting part. I got vaccinated last week. No idea which arm I'm in. And what he means by that is which arm of the vaccine trial. Though
I had trouble with that. I like, which arm did they put it in? You must know that.
Right. No idea which arm I am in, though, as it's double blinded, but I am either in group 2 or group 4 because I've only been asked to go in at day 28 and 182 days, optional 1 year after vaccination, plus 1 year after vaccination. The control group is given a vaccine that is for Neisseria meningitidis. In addition to this, when I was given my vaccination, the doctor told us that the group at Oxford has recently done a non-human primate vaccination challenge with SARS-CoV-2 with no evidence of antibody-dependent enhancement and good antibody titers. He had no information on what levels or what type they were when I asked. He said it's not often you get a microbiologist join the clinical trial. So this listener was really on the ball asking good questions. The trial has recruited the required sample size because when I had my vaccine pre-screening, they wanted to recruit everyone within 2 weeks across 5 different UK sites, then vaccinate on or after May 2nd. The recruitment links are actually closed now. The platform has been relatively well studied, and this is why they did not do an in vivo animal preclinical or phase 1 trial, but instead ran one in parallel. well studied, as in they have used influenza A antigens and tuberculosis antigens in previous human phase 1s and in vivo mouse models phase 1 for a MERS-S spike protein version. So far, I have no side effects, just the usual injection soreness in the arm. Who knows? I might have gotten the control vaccine. I've been filling out my vaccine diary daily as mandated, so hopefully I'm generating good data for the PIs involved. Stay safe and healthy. Brian, normally a PhD student, but currently seconded to COVID-19 PCR testing on 12-hour shifts.
So we should add that phase 1 has been in MERS model phase 1. I don't know what he means by that, but there's a phase 1 of the MERS ChAdOx in humans. We just mentioned that.
Right.
I don't know if he meant that or not.
Just reading from, we can even put a link to this clinical trial. This clinicaltrials.gov site is really good and probably understandable by almost anybody. So the official title of this study is a phase 1-2, that's combined phase 1 and 2, study to determine efficacy, safety, and immunogenicity of the candidate coronavirus disease vaccine, blah, 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 in UK healthy volunteers. And they're doing a single dose and a double dose, that is prime boost, with appropriate controls. The control being, what was it? The meningococcus vaccine?
Mm-hmm.
Weird. One of the things I wanted to point out is that the estimated study primary completion date and estimated study completion date is May 2021.
Mm-hmm.
Okay.
Mm-hmm.
Now, whether I don't know whether they can somehow beat that, because this is, I forget, somewhere in here are the details as to how long a trial actually lasts. But fast tracking is a relative term when it comes to vaccines.
I assume that these people are going to be monitored regularly to see who gets infected and who doesn't. I'm sure if they see some good numbers, they would consider ending it and just giving everybody the vaccine, right, including the placebo arm. Although it's interesting, in this case, they didn't actually use a no vaccine because they're using another vaccine. And so in both cases, you're going to get arm soreness. So,
yeah.
Right.
In the past, if you use just PBS, you don't get a sore arm, and then people know that they're in the control phase. That's interesting.
So they are going to sample at 182 days. So that's, 6 months in. And that's when I think the earliest we would expect there to be a change in the trial design.
So I told Doris Cully, who was on TWiV a couple of weeks ago, and she, I said, "What do you think about this combined 1-2?" She said, "These are unusual times. That's where we're doing all this." And I think that's true, because normally you wouldn't be doing all this. You would be doing it the usual way. But the vaccine is going to be the answer probably to getting us out of this. So. And
I think, understanding that this is being fast-tracked is important. That should heighten our vigilance. We want to make sure we don't do something bad here, because that's possible.
I wanted to, so thank you for that. That's very good from someone in the vaccine.
And he should, in any case, maybe get vaccinated. I mean, he will be vaccinated against the Neisseria.
Yeah, exactly.
or to chimpanzee adenovirus.
That's the booby prize, is the vaccination against Neisseria.
Right.
I wanted to point out a perspective in Science by Barney Graham, who has worked for many years on paramyxoviruses, in particular monoclonal antibodies. We did one of his papers a while ago on TWiV, and also vaccines against them. And this is called Rapid COVID-19 Vaccine Development. And he writes, his abstract is one sentence: finding the fastest pathway to vaccine availability includes the avoidance of safety pitfalls, which is important, right? Mm-hmm. And he says, vaccines can have two general kinds of problems. One is antibody-dependent enhancement, and the other is vaccine-associated enhanced respiratory disease. So we have ADE and VAIRD. ADE has been described for a cat coronaviruses. So the antibodies, it's like dengue, ADE. The antibodies do bind the virus, but they don't block infectivity, and they allow the virus to get into different cells via the antibody binding to an Fc receptor on the cell surface. So, he said, okay, this happens for dengue. We know it happens for this feline infectious peritonitis virus, but he said there's no experimental data showing in vivo that this type of entry is relevant to the pathophysiology of SARS-CoV-1, because it was reported that it might. He said, so we don't think it has anything. There's no evidence for SARS-CoV-2 either. But the other syndrome is VAIRD, and this happened in children in the 1960s when they were given an inactivated virus vaccine for both measles and respiratory syncytial virus. And so these antigens were incorrect, and they gave rise to antibodies that caused pathology. And so more serious disease after immunization. And he said that basically delayed development of a respiratory syncytial vaccine for 30 years, while people figured out why this happened. And he said it's important to make sure that this doesn't happen in any SARS-CoV-2 vaccine. He says judicious evaluation of candidate vaccines in healthy adults in parallel with vaccine studies in animals and coincident process development to scale up provides a path forward. So you got to be careful. He has a lot of experience in this, and he is. It's really nice, worth reading.
Yeah, this paper is a good description of those problems, which early on in this, I was conflating all that into one sort of vaccine-induced problem, but it's more complicated than that, and this helps sort it out. There's still no RSV vaccine, right?
So there's some experimental vaccines, including an RNA-based vaccine. But yes, there is no RSV vaccine yet, right? Monoclonals that you can use to treat this children, yeah.
I often, when people say, "Oh, there's going to be a vaccine in next year," RSV is often my sort of counterexample, because they've been granted under different circumstances and with evolving technology, but they've been working on that for 40 or 50 years.
Yeah, well, they had that bad experience, and so people avoided it, and for many years. And I met Barney Graham in January at a meeting, and
when you could still go to meetings.
When we could still go to meetings, and at this meeting, this was about enterovirus D68. And I went up to him and asked him a question, and he ignored my question, and instead he said, "I'm really worried about this therapeutic approach." And I'm not going to tell you what it was described at the meeting. He said, it took us 50 years to recover from a bad RS virus vaccine, and I don't want the same thing to happen. Mm-hmm. He really knows what happened here. So, okay, you picked an interesting story, Rich.
Yeah, this is a Science article cited as a policy forum that I think was sent to me from by one of my Brighton collaboration buddies. And it's called A Strategic Approach to COVID-19 Vaccine R&D by Lawrence Corey, John Mascola, Anthony Fauci, and Francis Collins. And Lawrence Corey is from the Fred Hutch Cancer Research Center in Seattle, and the Departments of Medicine and Lab Medicine at University of Washington, Seattle, a joint appointment. John Mascola is from the Vaccine Research Center at National Institutes of Allergy and Infectious Diseases. And, of course, we're familiar with Anthony Fauci and Francis Collins, who are respectively the head of NIAID and the head of the whole NIH. And this describes a public-private partnership and platform for harmonized clinical trials and how that aims to accelerate licensure and distribution. And this is a really nice summary of what is needed in order to get a vaccine, which they conclude is absolutely necessary to ultimately get us out of this mess. And I just highlighted a couple of things here that summarize it. It really very nicely describes the whole problem and vaccine strategies and everything else. So the goal of the paper is, they say, we discuss a collaborative program that has recently emerged that they call ACTIV, ACTIV, for Accelerating COVID-19 Therapeutic Interventions and Vaccines, a public-private partnership spearheaded by the U.S. National Institutes of Health, brings together the strengths of all sectors at a time of global urgency. They discuss a collaborative platform for conducting harmonized, randomized, controlled vaccine efficacy trials with the aim to generate essential safety and efficacy data for candidate vaccines, several in parallel, and accelerate licensure and distribution. And I thought the details of the collaboration was interesting. NIH has partnered with its sister agencies in the Department of Health and Human Services, including the Food and Drug Administration, Centers for Disease Control and Prevention, and BARDA, the Biomedical Advanced Research and Development Authority, other U.S. government departments, including the Departments of Defense and Veterans Affairs, the European Medicines Agencies, and representatives from academia, philanthropic organizations, and more than 15 biopharmaceutical companies, and the Foundation for NIH. Whoa. This forum allows for discussions and consensus on vaccine trial designs, rapid data sharing, and close collaborations between the public and private sectors to rapidly and efficiently conduct vaccine efficacy studies. Go for it. Sounds great to me.
Speaking of these partnerships, so the last TWiV, 612, was an interview I did in Singapore in December with the CEO of Ppv, a nonprofit that's trying to support studies on vaccines. And he has Ppv, I just realized this now, and I hadn't said it in that interview, but Ppv is funding Moderna's RNA vaccine work. So they decided to do that early on. So
and they're funding several others, about a half a dozen, I think.
Yep. This is exactly what they're meant to do.
So I've just been checking. You might remember, I think I told you I went to a Research America webinar that included Francis Collins and Keith Yamamoto. And Francis Collins described this active partnership, and I have screenshots of the various companies, and as Rich described it, all the various entities that are involved. Mm-hmm. So it does sound really good. One thing I wanted to mention. Oh, no, wait. It has to do with the next paper. Never mind.
So two more things. First, if you want a general overview of vaccines, you might want to check out my virology lecture on vaccines from the 2020 course, which covers a lot of these vectors and SARS-CoV-2 vaccines. So we'll put a link to that in the show notes. And then finally, I wanted to point out a story over at STAT, inside the NIH's controversial decision to stop its big Remdesivir study. So you may know, we've talked about this with Daniel Griffin, the U.S. coordinated trial of Remdesivir was found to be have a small effect on serious disease. And so they decided to stop the trial and give the drug to the placebo patients as well, right? Before the trial was over. And this article points out how this is a tough decision because, you want to make sure you collect the right data for your trial. On the other hand, if you show that a drug may be even a little bit useful, you want to save some lives, right? So it's like a conundrum. And for that, I think it's worth looking at.
This story described the data safety monitoring boards that I hadn't really appreciated and how they play a role in all of this. So they're abbreviated DSMBs. And I think both this story and the science story that Rich talked about are easily understandable by the lay audience, or at least TWiV listeners.
I was on a DSMB once for a clinical trial as an ad hoc reviewer, right? So I'm not permanent part of it. And I was impressed. They said to me, what do you think about what's going on in this trial? And I told them, and they listened to me, and they made them do something, right? And when people listen to people who know something about the virus, I think that's impressive. I'm losing faith in that. So that was a good thing. The last thing I want to just point out, which I found as I was looking at this stat article, there's another stat article, which is a quote of the CDC. 80,000 people died of influenza this past winter in the United States. 80,000 people. Mm-hmm. And that's the only article I've ever seen about this. Now, I understand many people are dying of something else. 80,000 people. It's a lot.
Yeah. It's as big as the big one that was several years ago. Yeah. They don't usually get up that high. Yeah. These are pandemic numbers. Usually it's less. And that's, around what SARS-CoV-2 deaths are in the U.S. right now. So that's quite interesting. Okay. Let's do some email. Rich, can you take that first one by
Mercia?
Yeah.
Mercia writes, I have a PhD, but not the right kind. Historian, not scientist. You have training in critical thinking. I would be fascinated to hear you take on the following paper by Li Lanjan et al. Gives a link. It's been extensively covered by the media, but I haven't found any good scientific discussion about the methodology. Of course, thanks to TWiV, I know that mutations aren't news. Of course, thanks to TWiV, I know that mutations aren't news. But the finding about substantial changes in pathogenicity has been interpreted as putting meat on the bones of the different strains theory. For example, why Italy and New York got hit higher than Washington or California. What say you? And that's right. This is a family show. So I can't say entirely what I really think about this, but I would say that their claims are at very least exaggerated, if not bogus. So this is now we've discussed this elsewhere before.
Yeah.
Is that right? This paper? TWiV 54.
Yeah.
Okay. It's titled Patient-Derived Mutations Impact Pathogenicity of... wait a minute. Let me not just gloss over this. Patient-derived mutations impact pathogenicity of SARS-CoV-2. That's the title. And basically, it describes, as we have described many times before, that if you look at the sequences of viruses that are coming through this, there are mutational changes that show up. Okay. And these guys take several of these and compare their growth in cell culture. And they notice some changes in viral RNA replication, not live virus, in cell culture, and they extrapolate that. They interpret that as an impact on pathogenicity, which is like false. Okay, it could be that some of these have a replication advantage relative to others in cultured monkey cells, but that has no bearing whatsoever on what they will do in humans, in particular, what their disease potential is in humans. So I would say that all this paper has is a couple of new mutants in it, and I would throw the rest out.
I also want to point out that they measure virus reproduction by PCR, and they don't do an infectivity assay, which seems inexcusable to me. You can do a plaque assay with this virus. And so, if they say, oh, this mutation causes an increase in RNA pieces in cell culture. What does that mean? We don't know what it means unless you're looking at infectious virus. So I agree that this probably won't see the light of day in its present form in terms of a journal. I hope not.
Right. This is, once again, This Week in Evolution, TWiV 54. You can just Google for that. And the title of the episode is Rough Drafts of SARS-CoV-2 Science. And so Nels and Vincent do a good job of talking about why these are only rough drafts.
And then our latest episode on TWiV was called From Soup to Nuts, where the nuts doesn't mean dessert. But Nels was afraid that was too subtle. The next one was from Amy, who says, I keep hearing experts say things would improve until we have a vaccine, but I don't hear experts talk about effective therapeutics in the same light. If we found a therapeutic that reduced mortality rates and reduced the amount of time they spent in the hospital from 2 to 3 weeks to a couple of days, couldn't that mean we would significantly free up health system capacity? Could this also speed up herd immunity in the event that the immune response is protective long enough to prevent reinfection for a helpful period of time? So, the problem with therapeutics, in my view, is that who are you going to give them to? If you wait till someone is really sick and they can't breathe and they're in a hospital, by then the virus titers are already dropping and it's not at all clear that an antiviral will make a difference in their survivability. Now, that was shown in the Remdesivir trial. You get a little bit of a benefit, which may be worth using, in the big term. You may save a lot of lives that way. That's great. But it's not like you feel like you have flu-like symptoms. You go to a doctor, they do a rapid in-office test. They give you a prescription for Tamiflu and you go take it. We don't have that for this. So I don't know that the antivirals are going to be all that useful. That's why I think the vaccine is really the key. I don't know what you guys think.
I would agree with that.
Yeah, I would agree. Getting the timing of any antivirals right is what's crucial. And unless we had an instant, really reliable test for if you're infected, it may be that they're going to be too late. And that could be why the Remdesivir trials don't look even as great as they might. People are saying, oh, you're just shortening the hospitalization time. Well, that could be important. And it could be even more important if the Remdesivir were given at an earlier time. I remember seeing somebody pointing out that, for now, certainly until we have a vaccine, a lot of these things are important from the standpoint of sort of a Moneyball philosophy. So that movie about the Oakland baseball team guy who figured out if you could make all these changes, then the team could succeed better. And it's kind of, if you could have Remdesivir, if you could, do certain other things that people are trying, that maybe in combination, those together can give us a strategy that helps until such time as we have a vaccine.
Of course, the other problem with Remdesivir is it's intravenous, so you have to go to a hospital to get
Correct.
it. So it doesn't help.
Correct.
But I do think you got to try all these things. You got to try therapeutics. We're doing vaccines big time. We'll see what works first.
It has occurred to me that, given that the major problems seem to be immunopathology of one sort, or at least immune stimulated pathology of one sort or another after the infection has peaked, that the treatment approach should focus on the immunology, which I think to some extent it has, rather than or in addition to the virology.
Yeah, agreed. All right, Kathy, can you take Lewis?
He writes, Dear TWiV, I saw some news coverage of this study published in The Lancet and thought it might be of interest for TWiV, given discussions of risk to frontline health workers in recent episodes. And he puts in the link, COVID-19 infection rates are no higher for NHS staff treating patients than in non-clinical roles. So NHS is the medical system in the UK. And he gives a link to the study. I haven't looked at this, but it seems like we may have talked about this before.
I think we did. Yeah, I think Alan in particular has mentioned this, that there's no evidence that they are any more at risk or sicker. What's the word I should use? Not more at risk. No higher infection rates. Right.
Mm-hmm.
So there's an article from the Popular Press and also a link now to the Lancet study.
Okay, Rich, take the next one.
Anonymous writes, hello, professors. I love you all and would like to remain a long-time anonymous admirer. One of my housemates, 25 years old, believes she has already had COVID-19 and so is immune. She's not been tested. 2 days ago, she visited a friend around the same age who was admitted to the hospital for severe GI distress. Her friend was tested for a handful of things related to this, related to his gut, but not for COVID. And she visited with him the day of his release and the day preceding the night he was admitted. This housemate did not tell me or our other housemates of her visit until sometime after she'd returned from seeing him outside the hospital, and me, in fact, in the kitchen where we were both unmasked and I was batch cooking some gruel to survive another week working at home. I asked her to let me wrap up alone for about 10 minutes, but I had to leave the same way she did to shutter myself in my room before waking early the next morning and carpet bombing all the kitchen, bathroom, and door surfaces with 10% bleach. So my question is, if my housemate is, quote, immune due to a previous infection, but comes into contact with someone actively infected, can she still shed infectious droplets, not talking about fomites, from virus trying unsuccessfully to replicate in her respiratory tract? I would say, in answer to your question, no, highly unlikely. I think if she became contaminated by interacting with this person, she could serve the function of a shopping cart and a fomite, and transmit some of that stuff, vector some of that stuff around. But if she is in fact immune, and that's a big if, and we don't really know for sure what immunity means in this circumstance, but if she is in fact immune, she should not replicate virus to any significant titer. And my sense is that that's pretty much a prerequisite to shedding it significantly elsewhere.
I think the issue is whether immunity is sterilizing or not, right?
Yeah.
And we don't know. It could be that you shed enough, even if you're immune, to infect others. In fact, with the common cold coronaviruses, that's the mode, right? You get infected, your immunity declines within the year, then you get reinfected, and it spreads that way. So I'm not sure. I think you could spread it. I think we can't say that you would not.
This also calls up, I was going to say, this calls up the question too of, she believes she has had COVID-19.
Yeah.
Of
course.
She could have been one of those people who had the flu. We just talked about how it was a high flu year. And so you just don't know. I
think in a situation like this, one has a responsibility to, to your housemates, okay, to be upfront about what you do. That's the whole, that's why we wear masks, not for us, for everybody else. This is not about you. It's about everybody else.
Martin writes, following on from Michael's comment about robotic dentistry in TWiV 611, I wonder how much of Dixon's whiskey they gave this female patient to calm her down before this groundbreaking operation. He sends a link to an article in Dentistry Online. Robot dentist in China is first to fit implants in patient without any human involvement. Okay.
And I'll tell you, the picture of this robotic thing is scarier than a dentist by a long shot.
Indeed. It's very scary. Thank you for sending that, Martin. Kathy.
Okay. David writes, a few TWiVs ago, an email asked about testing sewage for virus as a way to monitor for SARS-CoV-2 spread in communities. And you commented that it would be an interesting thing to try. Looks like Michigan is taking you up on the suggestion. And we have also talked about a number of studies looking at being able to monitor viruses in wastewater, including from investigator here at the University of Michigan. But this study that he sent involves Michigan State. And again, I'm waiting for the link to open up, but it's Poop Sleuths. MSU studies Detroit wastewater for next coronavirus outbreak. So it gives a link to that.
So it says they're doing it. We don't know any results yet. So maybe we'll find out in the future. That would be cool. Thank you, David. Rich.
Okay. We have, is this
another David?
Another David.
Mm-hmm.
Hi, all at TWiV. Thanks for all you're doing to keep keeping us at the coalface up to date with the current pandemic. Very much appreciated. I'd like to raise an issue which is of concern around how many people understand how to use diagnostic tests, especially politicians, but also many healthcare professionals and advisors. I wonder if you could help explain this further to your listeners. There are lots of discussions around sensitivity and specificity of tests, and many see this as the only important characteristic of a test, but it is not. The main utility of a test is the positive and negative predictive values, Ppv and Npv. Ppv is the likelihood that a positive test represents a true positive test, i.e. the person tested has the disease, and Npv is the likelihood that a negative test is a true negative, i.e. the person doesn't have the disease. Ppv and Npv are based on the sensitivity and specificity, which stay constant for a test, but are influenced by the prevalence of a disease in the population being tested. In a setting where the prevalence of the disease is high, a test will have a better Ppv and a worse Npv, whereas when the prevalence drops, the Ppv becomes worse and the Npv improves. Let me put this in the context of COVID-19 with a PCR test that has an estimated sensitivity of 80% and a specificity of 98%. We still don't really know these values, as no one has yet defined a gold standard test to compare against, but let's use these approximate values. If we are at the peak of a pandemic and say that 50% of those tests do have COVID-19, then the Ppv is 98.6% and the Npv is 83.1%. That is 97 to 98 out of 100 people with a positive test do have COVID-19 and 2 to 3 do not. 83 out of 100 people with a negative test do not have COVID-19, but 17 actually do have COVID-19. This means that we can rely on a positive test and cohort patients together, as the risk of putting a patient who is actually negative in with those who are positive is low. But we cannot put all the negatives together, as there is actually a lot of infected people in that group who could still infect others. What happens when we start to control the pandemic and the prevalence drops to 5%? In this situation, Ppv is 67.8%, Npv is 98.9%. We now have the opposite problem. We can be sure that those who test negative do not have COVID-19 and therefore can be mixed together, but 32 out of 100 of those who test positive now actually do not have COVID-19, and if we cohort them with the other positives, we will still be at risk for becoming infected, and it gets worse as the prevalence drops further. The basic upshot of all this is that we have to adapt how to use diagnostic tests in the all of the time, because Ppv and Npv are changing all of the time, and our national and local infection control policies need to reflect this. So we are now in the situation where we are ramping up the testing in all of countries and starting to test thousands of asymptomatic people who will have a very low prevalence of COVID-19 with a PCR test, which will give lots of false positives, leading to screening and isolation of thousands of false contacts and potentially giving false reassurance about past infection and herd immunity. It's a case of wrong test in the wrong situation. Hope this makes sense. If not, then please just use this idea for a wider discussion, as I think this is an idea of diagnostic testing which many do not understand. Keep up the great work, David. And David is a medical doctor and clinical microbiologist in the UK. I would have to read over this another 10 times to fully assimilate it. But the take-home message that I get is that just the sensitivity of a test and its selectivity are sort of raw fixed values, but their meaning can change depending on the dynamics of the infection. And this, positive and negative predictive values are the important points. And wow, I was totally unaware of this.
Mm-hmm.
Yeah, the problem arises is when you have high prevalence or low prevalence, and then it impacts on how you want to put people together. And that, many people have said, why don't we take all the at-risks and just let them stay home, right? But if one of them is infected and you don't know it because of a bad test, that could be bad, right?
Right.
But he says, and this I don't get, we're ramping up testing, thousands of asymptomatic people. So that's a low prevalence situation. We're going to get a lot of false positives. And so he's saying we shouldn't be doing this, but I don't see why.
Well, I mean, I think you can do it, but you just have to know what the data mean out the other end.
Well, if you have a positive, it could be a false positive, but in either case, you want to contact trace and quarantine. And at the worst, you've inconvenienced a bunch of people for 2 weeks, which I don't think is a bad outcome, right?
Right.
Anyway, that's my interpretation of that. Well, anyway, I appreciate the explanation. We've actually encountered NPV and Ppv before, but this is a good explanation.
Mm-hmm.
Ted writes, greetings. This is my first virus with your podcast. Excellent work. I have heard, no, it's not. You've had many viruses. So maybe with our podcast. I've heard some of your comments about transmission risk differences with joggers, bikers, and even barbershop singers. As we start to open up, how should we address these distancing for practices and gatherings with singers and groups of singers? Okay, Kathy, you must have thoughts on this.
Well, I pitted a lot of information because Rich was the one who mentioned that he was going to go to this webinar, and so he can tell us about that. The webinar's title is A Conversation, What Do Science and Data Say About the Near-Term Future of Singing? And it was presented by a number of choral director groups and the Barbershop Harmony Society and so forth. It's 2.5 hours long. And I think one of the things that impressed Rich was that it included a number of experts, including Donald Milton, who is the developer of the Gesundheit 2, which we talked about and is the show image on the webpage for TWiV 480. So that was cool. So Rich can expand a little more about the webinar if you want.
So, it was a webinar, but I really wanted to sort of really crash in and say, I've read your papers. I've read your papers. The Gesundheit, for those who don't remember, the Gesundheit 2 is a machine that's like a giant cone that fits over your head that you can breathe or sneeze or cough into. And it not only collects what comes out, but sorts it into different sizes, and so it measures what's coming out of your lungs, and actually can assay it and ask whether it's got virus in it, et cetera. He's done this with flu patients, and et cetera. As I told Kathy, the webinar was long, and part of it is because you can't make these scientists shut up. They also had an otolaryngologist on the show who's also a singer. And the bottom line was that there is good evidence for, you got to be careful with the vocabulary here. There's good evidence for aerosol I don't even know if I'm using the word right. That's why I'm hesitating. Aerosol transmission of the virus. Not just over short distances, but even longer distances. Of course, this is going to quantitatively change with distance and et cetera, but the experience with singers and choirs, which Kathy can comment on some more, suggests that this can be a problem. The bottom line that I got from out of the webinar that I communicated to our chorus, it's a barbershop chorus that I participate in, is that there's just no risk-free way to do this. All right. And not only that, but getting a choir together and singing is one of the riskier group practices that you can do, because we're, expelling air and all of its contents from our lungs with some force, standing close to each other. So basically what we've decided in our chorus is, sure, we'll pay attention to the recommendations that come out of our community and the world of scientists at large. But we all have to understand that if we have a rehearsal and somebody comes in and is shedding virus, there's a significant risk that's going to get spread around based on experience, to several members of the choir. And there's not much that is practical in a chorus situation that you can do to prevent that. So everybody going back into this is going to have to understand and accept that risk. And my guess is that for us, from a practical point of view, it's going to mean that it'll be a while before we actually get back together again. And when we do, I can imagine that some of our more at-risk people are just going to stay home. I would even recommend that they do so, which is too bad, because a lot of those people are old timers who've been around with the chorus for a long time and make real contributions.
So, as I said, I haven't watched the webinar, but it sounded like it was too devastating, and I just wasn't going to be able to handle it. I was unaware of a major story that in the choral world that came out March 29th in the LA Times about a rehearsal in Washington State of a community choir, about half of whom were present at 2 different weeks of rehearsal. And I was consulting with another group in Arizona, and someone in absentia from that group weighed in later. Particularly, he pointed out this MMWR report. So I am really grateful to Mark for that. He turns out to be a TWiV listener as well. And his comments from this MMWR report, which I have read, the index case had symptoms at the choir rehearsal when the virus was most likely spread on March 10th. That was the rehearsal. And March 3rd rehearsal, that person had no symptoms. The choir members were seated 6 to 10 inches apart, not the recommended 6 feet, and there were multiple intermingling opportunities. And the report concludes that the transmission took place through droplet and fomite transmission, although it does mention briefly the potential for emitting aerosols when singing. I didn't prove that transmission by aerosols didn't take place, but it makes the point that droplet and fomite transmission were likely the primary modes of transmission. So the people afterward put chairs together. The average age of the people in this chorus was 69. It doesn't mention anywhere whether people hugged at the end of the rehearsal or before the rehearsal, any of those kinds of things. But the person had started feeling ill on March 7th. And if he had not attended on March 10th, if people had remained 6 feet apart, if they had not touched fomites, if they had worn masks, probably they didn't, because this was so early in March that people weren't wearing masks then. Maybe the outcome would have been different. So I came away a little bit less devastated having read the MMWR report and Mark's analysis of it. But I agree with Rich. I think for the near term, we're not going to go back to singing in choruses. And this is really devastating for a lot of people. It's a major community socializing kind of thing. I think I may have said it on TWiV, but I know I've said it numerous times that, I can be having a really lousy day, and then I go to a choir rehearsal and it completely changes my mood. And so, it's going to be tough to give this up for a long time.
This figure is really striking in this, with all the people the index case up in the upper left, and then everybody else except the front right, the farthest away, I guess, were infected. Isn't that amazing?
So is this a figure in the MMWR? Because I thought it did not include the maps.
Yeah.
Oh, I see it now. Okay, I didn't see that the first time. Right.
Which I think is now a promotional poster by CDC.
Okay, so this may not be the actual map because they said they did not include that because it was going to be too revealing, as it were. So
but
they have them
like, but you don't know who's who, right?
Correct. But so this is Anyway,
it's the poster is quite striking.
Yeah.
Well, I guess you can't have masks if you're going to sing. That would be silly, right?
Yeah.
We discussed that, and that's just not gonna work.
I don't necessarily agree with that, but again, maybe I'm just trying to see it as half full and not half empty. I think you could sing with a mask if it meant
Kathy,
that or not at all.
Is your chorus doing anything in the meantime, like any sort of virtual gathering?
No, I think in part it's because the school year is still going on and our director has a lot of, potentially school year things, responsibilities. We usually have a open to all summer sings and those are canceled, and we'll just have to see what happens when we come back, when we come to September, what the situation is.
Our situation is probably, quite different than you. They're different sorts of choruses, but, our active membership is only about 30 individuals, and we ordinarily rehearse once a week for maybe 2.5 hours. And basically we've gone to Zoom meetings. Mm-hmm. And you can't really do any significant singing, though we do. We have people sing their part a cappella in smaller gatherings, Zoom rooms with other individuals. So we're working on a particular song. But we've also taken this as an opportunity to do some other things like music theory. And listening to other performances and evaluating them and that kind of stuff. And at very least, it keeps us in touch with each other and keeps the motivation and the social aspects of it going to some extent. And actually has some, like the music theory stuff has some advantages. It's stuff that we wouldn't do otherwise.
Right. There are a number of virtual performances that people have seen. A really good one from a Canadian group that's For the Longest Time, and I think somebody could make a really good impact if they could develop software that was really amenable to people singing together. You've been on a Zoom or a Blue Jeans or anything where the transmission just freezes, and that kind of asynchrony would not work. And so it would be great if somebody could figure out how to do that.
I'm on a call today, and I was on the similar one 2 weeks ago of theater owners who want to, live action plays and stuff, musicals where they want to know. And I think it's the same issue because they're spitting. Mm-hmm. That's funny. I made a comment the last time. I said, these actors spit a lot as they're talking loud, right? And one guy said, yeah, especially one who we will not name. So apparently there's someone who's infamous for spitting a lot.
I think there's a lot of people who are famous for being spitters.
I'm pretty close to the whole theater thing because of my daughter's profession. Yeah. And it's a problem, not only from the point of view of people getting together, but audiences. I mean, theaters actually don't even make budget with a full house.
Right.
Okay. They need donors. So the notion of trying to run a show with a limited to 25% capacity just is not going to fly.
That's
and so
exactly what the guy said. Yep.
much the same as laboratories and that kind of stuff. And in the university where Sarah, so the theater's shut down, and in the university where Sarah works, there's a big quandary about can they what's going to happen in the short term, at least, with their theater program. What productions are they going to put on? When are they going to do them, et cetera? So it's a big problem.
Kathy, you were up next.
Amelia. Amelia. I just saw this article regarding COVID-19 sniffing dogs. Please, oh please, discuss. And Amelia sends a link. I wandered around the internet following the scent of this topic, for a little while, and ran into another fantastic article about the scent of Parkinson's disease. What are your thoughts on detecting disease in general and a viral disease in particular? And Amelia gives the link to the American Parkinson Disease Association that has the links to the ongoing research. And I haven't looked at these, but Vincent has a comment.
I mean, the article, the dogs are not sniffing out the actual virus, of course. The patients have a particular scent, I guess. It's certainly the case for Parkinson's, and dogs can detect those people. And so it must be that perhaps Parkinson's causes different organic vapors that you admit to be slightly different. So the same could be for COVID-19 patients, I suppose. I don't know how long it takes to do this, to train a dog to distinguish. I don't know how symptomatic you have to be. There's so many questions here that are interesting. But it would be interesting if you could do this. It would be interesting if you could sniff out an asymptomatically infected person just by virtue of, the virus is reproducing and making changes, organic molecules. When plants are infected by certain viruses, they give off different organic vapors that attract aphids. And we've talked about these papers a long time ago on past TWiVs. Very interesting stuff. In fact, the virus is inducing these vapors to attract aphids so that they're they're not doing it on purpose, but this is how it's evolved to be able to attract aphids, which will spread the infection. So the idea that a virus is making a change in the scent is not crazy. So I think it's fascinating, but I don't know if it takes a long time. Maybe this will be over by the time they get the dogs. But, you can sniff drugs, right, with dogs. They do that all the time at airports.
Yeah, this it wouldn't surprise me at all if this were a possibility. I mean, can you imagine having a dog's sense of smell? It must be,
torture sometimes.
What is their reality like? It must be another layer of perception that colors everything else that they do.
They're always
fascinating.
They smell everything. Yeah. When they go outside, they're always like, "What are you smelling? This little square inch? What do you smell there?" So I think Dove mentioned this once. So that it's like a kaleidoscope for them. All these smells. Yeah.
There was a program at UF where they were training dogs, and I've seen this work. They train dogs to sniff out bed bugs. So they can go into a hotel room and tell whether there's a bed bug contamination.
I'm always amused when I'm standing at a luggage thing in an airport, and they bring the dogs and they sniff my carry-on or something. It's so funny, but that's how it works. Rich, you're up next.
Asal writes, "Hi TWiV team, your productivity is admirable. I'm a TWiV core listener and have been to 2 TWiV recordings at 2 ASV events so far. As a molecular diagnostic technical consultant who has performed and overseen many viral load assay operations, I would like to point out what I have been hearing from clinicians and even scientists about the relationship of CT value to viral load in regards to SARS-CoV-2 positivity. Oh, good. I want to hear this. In a non-emergency world, a quantitative PCR for determination of viral load takes more than a simple relationship between a CT value and the number of viral particles per unit of sample. These assays sometimes take years to be developed as accurate predictors of viral load and years more to establish a conventional international unit for a single strain of any virus. As an example, there were many controversies before CMV was agreed to be tested and counted off the plasma versus whole blood. There is yet to be a consensus for EBV in this matter. That aside, for establishing a predictive standard curve, a lab needs to test many certified quality materials with known viral load in serial dilution and in replications. After linearity is established, it will need to be evaluated at least every 6 months and reestablished at the first signs of trends or shifts in the quantitative QC values. Mandatory, graded, and frequent proficiency testing is in place so that labs using the same platform can compare their obtained results with their peers nationally. Even then, clinicians are advised to stick to one platform and preferably one lab to monitor their patient's viral load throughout the treatment course. We are far away in calling any PCR test currently performed a quantitative PCR for SARS-CoV-2. The CT of 13 in one lab could be 20 in another lab due to many known and unknown variations. Bottom line is that it takes more than a CT correlation to call an assay quantitative. What can we do in the meantime for our SARS-CoV-2 is to call the detection with a low CT, a strong positive qualitative, and one with a high CT value, a weak positive, and avoid calling them high or low viral quantities because we haven't trialed enough to mathematically establish the numbers in correlation with CT values, fluorescent units, and other components of the calculation. Even that gradient interpretation should be affected or changed from lab to lab and between different assay platforms. I'm writing to you from my backyard in Colleyville, Texas. It is an after rain 22C beautiful spring weather. As always, I'm very grateful for living in the same time with y'all Microbe TV co-hosts and learning in almost real time from you and your guests. Well, Asal, thank you. It goes both ways because this answers many questions that I've had. I mean, I thought it should be straightforward with a standard in a test to get a quantitative picture from a CT determination from PCR. But apparently, it's a lot more complicated than that, and I appreciate these insights.
I assume you also need to use infectious virus to calibrate this, right? If you really want to talk about virus load, you can't use just, RNA, known amount of RNA, because you don't know. That's why it's called viral load and not RNA load, right? Mm-hmm. So do you think we should say briefly what a CT is in case people don't know what that is?
We tried the other day, and I kind of fell flat on my face. Why don't you give it a rip? Actually, I went back and looked up the quantitative PCR assay. I don't think we need to go into that kind of detail, but a CT makes sense.
The simple answer is it's a quantitative measure of cycle threshold, right? Of how many times, how many cycles you need to go through before you can detect your product coming from your input template. So your experimental sample goes in as the template, and how many rounds of cycling do you go through before you can detect that it's been amplified?
So you're doing a amplification by PCR, and at every cycle of amplification, you can measure how much product is made because you have a fluorescent dye, for example, that gives you a signal, as opposed to a conventional PCR where you do the whole thing and you look at the end at a band on a gel. So every cycle you can measure how much product you have, and you can set a threshold, which is the CT, for saying, in this test you need a CT of this, and people have thrown this CT around like 13. People say, it's 13, there's a lot of virus. That's what Ian Lipkin said, because if you get a signal early on in the 13th cycle, it suggests you have more virus.
It means you started with a lot of material, so it didn't take you many cycles to get to the threshold.
Right.
But all that, as this email beautifully points out, all that has to be calibrated. Right. And so thank you for that. I just forgot what CT it meant.
Me too. and I was thinking about the fact that, we've had samples and shared them with another lab and done the same PCR, and their CT values for the same samples are a little bit different from ours, even if we're using the same PCR machine. Exactly. So, yeah.
All right, let's do one more, as I have a 1:00 meeting, sadly. Manoy writes, TWiV 604, you're talking about OPV. You talked about stimulation of innate immune system responses as a possible mechanism that works when using OPV to prevent infection. Well, that's the theory. No one's tried it for SARS-CoV-2 yet. That made me think about why the infection numbers are low in countries like India. I believe innate immunity is higher in places like India. I grew up in India, but after 30 years of living in the U.S., when I visit India, I need to be careful with what I do there. My brother-in-law, who was born in the U.S., will have to be even more careful by getting additional vaccines and drinking boiled water, etc. So it seems to me that people in India generally have higher immunity than in the U.S. What are your thoughts? Does this explain the lower numbers of COVID-19 cases in India? P.S. I'm only an armchair virologist. I have no expertise in biology. Pardon my ignorance. P.S. 2. No, I don't get any extra credit for having it answered, although I probably will get bragging rights with my microbiologist wife, Deepa, and my geneticist sister, Binu. That's good. No.
So this reminds me of, I'm pretty sure it was a TWiP This Week in Parasitism episode with Daniel and Dixon and you, Vincent, where Daniel reported about somebody who had grown up in a country and then moved to the United States and thought that they would be protected from whatever the parasites were when they went back to that country, but they weren't. And so that seems to be one of the factors that could be at play here.
Yeah. Well, I'm just worried that we really don't know how many cases there are in any country. Well, particularly India. I'm not sure how much testing is being done, right?
Yeah, I think testing is more likely to be I certainly wouldn't draw any conclusions based on case numbers until I understood very clearly what the testing capacity was and who was getting tested, et cetera. If I'm not mistaken, that this is the same kind of argument that was used to draw a possible association with BCG immunization.
Yeah, of course. Exactly the same. Yeah. It is the same. And I mean, there are lots of infections going on in India for sure, and that could impact it. But that's about all we can say until someone controls for the testing and also does a trial to address it, right? Same with BCG, right? They think in countries that use BCG vaccine, there's less SARS-CoV-2, but now they're doing an actual trial to see if it impacts it. So I don't know what you would do in India because there's nothing, right?
I want to respond to his PS1 where he says he's only an armchair virologist, no expertise in biological sciences. Pardon my ignorance. I would say that if you are aware of your ignorance in some area, there is no reason to feel like you need to be pardoned.
No, it's the same.
Understanding the limits of your understanding and seeking to understand better is what it's all about.
Isn't there a famous quote about that? Knowing the limits of your knowledge. Well, I don't remember what it is.
We'll find it.
This feeling that there's a cool quote. Okay, that is TWiV 613.
As usual, we'll put these letters in the show notes, and as usual, we've made it through about 10% of the pages of letters.
Like Rich said, we talk too much, right?
Yeah, but what the heck?
That's what we do. We will get through these letters because we have a lot of time. So stick with us. Microbe.tv/TWiV is where we will have links to these articles and letters and so forth. If you want to send us a question, TWiV@microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Nothing like talking about science to get me less grumpy, frankly.
Yes, me too.
About you. In our pre-show, we were all very, well, not
I wasn't grumpy.
No, you weren't grumpy. Kathy and I were very grumpy, but I'm calmed down now. So talking science is good. And for Kathy, it's that or being in a choir, right?
Exactly.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, I found a quote for you.
Let's see.
This is Socrates, and it's a little convoluted. I am wiser than this man, it is likely that neither of us knows anything worthwhile, but he thinks he knows something when he does not. Whereas when I do not know, neither do I think I know, so I am likely to be wiser than he is to this small extent, that I do not think I know what I do not know.
So that's the sentiment.
I love it.
I thought there was something more concise, though. But Socrates, like scientists, like to talk about. I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.